You earlier asked about the concept of the research, how this is being done. And of course, we recognize the mint family for their flavors. Now, if you are interested in finding out how those flavors emerge, you can go in and search for those genes. Now, what Abby said is, should you have a genome at hand, you can drive the discovery of these pathways in reverse. You have the genes, and then you can find out what kind of chemistry they make without the prior knowledge, without the prior need really to learn about the chemistry. Now, why are we interested in learning about those chemistries? And I just want to take 2-3 sentences talking about the reason behind that. We are excited about learning how plants make these chemistries because this would allow us to use these pathways in heterologous hosts. And you earlier mentioned yeast. Yeast is one of those hosts where you can put these pathways in, and then you can use yeast to make these chemistries. Now, we call this branch of science, we call this synthetic biology because it's sort of at the intersection between chemistry, between biology, between bioinformatics, and everything that Abby has talked about. So I think that synthetic biology is a key concept because then we don't have to go out into the wild. We don't have to forage plants that might be at the edge of extinction. We can use this knowledge in these modern times in biotechnology to produce these compounds, these molecules.
That's fascinating. So just to make sure I understand what you're saying that we can almost propagate these compounds using something like a yeast or for example, and that way we don't need to just rely on what's growing in nature, because obviously we're living on a planet that's undergoing some very extreme, climatic changes. And obviously that is going to put some strain on some of these ecological niches in which these plants are found. And so it seems to me that this is, you mentioned earlier that, perhaps something that we brought up was good timing. It seems like this is great timing then for us to have these computational approaches like Abby, the one that you're using, so that we can go in and grab this knowledge and then hopefully using sort of the bioinformatics and some of these more emerging practices, create them without having to steal them or be, like you mentioned earlier, biopirates.
Yeah, and another side of that is that we can also avoid using synthetic measures to make these compounds. So a lot of like pharmaceuticals and stuff will make these compounds synthetically. And that can often lead to a lot of waste products. These reactions are often pretty low in efficiency as well. So there's it's expensive. It can make some toxic compounds. It's not very good. So if we can use yeast or other plants or whatever other system to create the molecules that plants are making within themselves that are helpful to humans, then we can increase the efficiency, especially with like yeast. The the stuff that you would get coming off of that, like it wouldn't be toxic. It would be something that you can use for other means. You can use yeast as like feedstock or whatever. There's a lot of other applications that something like that, it's a lot less toxic. It can be more efficient. So it also helps in the fully synthetic route to be using the machinery of the plants or of the yeast.